Hello everyone, I'm Mr. Furlong, and today we're talking about changes in the signal transduction pathway. In the last video, we looked at how the signal transduction pathway works, and any kind of change in that pathway will alter what the cell's response is going to be. So if you recall, we have a ligand that attaches to a receptor, which we'll have here in blue. The shape of the receptor has to match the ligand, which then causes an alpha protein to head on over to another protein. It could be an enzyme that causes ATP to become CAMP, a secondary messenger, which is then going to cause some type of a cell response. But anywhere along this pathway, something could change the way that the cell is going to respond. For instance, there might be a mutation in the receptor protein. So for example, take a look at how the receptor changes. It's not the same as the ligand anymore. When the ligand goes to attach to the receptor, it doesn't have the right shape, and so nothing else is going to happen after that. The next step is not happening because it doesn't have the right shape. Hence, the cell's response is not going to occur. Now take a close look at our orange protein and watch if there's a mutation in that protein, and we have our ligand which can attach to the receptor. That's going to cause our alpha protein to move over, but because this enzyme doesn't have the right shape because of a mutation, the ATP is not going to change into CAMP, hence no cell response. Now we know that chemicals can interfere with any part of the pathway, and these chemicals could actually, though, either inhibit the pathway, like we've been showing, or it could actually activate a pathway. So for instance, here we see the fibroblast growth factor proteins, and right now they're in an inactive form. But when we have the fibroblast growth factor attaching to them, notice what happens in this other diagram. They move closer together. This is actually going to activate a pathway that allows for the growth of fibroblasts. So in one example, the chemical inhibited a pathway. In this example, it will activate a pathway. But it gets even stranger than that. So for instance, with aspirin, can actually have a reaction with the amino acid serine. Now depending on which enzyme is present, these enzymes are called cyclooxygenase, and there's two of them. There's what it's called COX-1 and COX-2. That if COX-1 is present, that enzyme will actually be inactivated, which will prevent the aspirin from doing its job. But with COX-2, that will actually switch on another pathway that will produce a protective lipid mediator. So here's a case where same chemical, depending on what enzyme is present, will cause either an inhibition of the pathway or it will activate a pathway. Lots of crazy stuff going inside our cells, folks. We'll see you in class.